Hello students. Today we can start the class with a question. Have you ever wondered that we look similar to our parents? All organisms in this world, they look similar to their parents. Why? What is the reason? We have the same eye color, hair color, and our height may be also be similar to our parent, and some of the actions which we do it will be also similar to our parents. Have you ever seen this? What comes in your mind? What makes us similar to our parents? So in this video, we can discuss about that. So the reason behind this is DNA. DNA, it acts as a blueprint guiding our development. So between any two individuals, the DNA is 99.9% identical and 0.1% gives variation that influence our uniqueness. How can this one single molecule contain so much mystery and wonder? We can discuss this in detail in this session. So, life is spelled as ATCG. So, in English, we have the alphabets, no? A, B, C, D, like that. DNA has also the alphabet. We call it as ATCG. So, it gives the information. It brings the information from a parent to the offerings. Now, before learning about the DNA, we should know where this DNA is located. So, DNA which is present inside the nucleus of an eukaryote and which is seen in the cytoplasm of the prokaryote, we call it as a nuclear DNA. Apart from this, DNA can also be seen in some cell organelles. So, here mitochondria also has DNA. So, we call it as a mitochondrial DNA. It helps in the function of mitochondria. It supports the function of mitochondria. Likewise, the other cell organelle, chloroplast, it also contains the DNA. So, it supports in the function of the chloroplast. So, it is also called as chloroplast DNA. In humans or in eukaryotes, we can see nucleus in the center of the cell, and inside the nucleus, we have the chromosomes, and the chromosomes only we call it as DNA. Okay, so this is the blueprint which carries the information from our parents. So the DNA, it is a double-stranded one and it has the sugar phosphate back and the nucleotides. These are the alphabets. So the arrangement or the order of these sequences of this bases determines the information available for building and maintaining an organism. Now, what is the DNA? Because it contains all the mystery and wonders, no? So we can learn what is DNA. DNA, it stands for deoxyribose nucleic acid. It is a molecule that gives instructions for an organism which needs to develop, live and reproduce. And these instructions are found inside every cell and they are passed from their parents to their children. So in every person we have the same DNA. That only I told. In between two persons, the DNA will be 99.9% identical and 0.1% variations give the uniqueness to the individual. And the DNA was first discovered by Watson and Crick in 1950. Structure of the DNA molecule, how it is made. So DNA, it is a very large molecule. It is made up of a long chain of subunits and these subunits we call as the nucleotides. So each nucleotide is made up of a sugar called a deoxyribose, a phosphate group and organic base. So in the sugar, it is of two different types, ribose sugar and deoxyribose. So DNA comes under the deoxyribose group. So what do you mean by ribose? It is a sugar molecule only, similar to of a glucose, with only 5 carbon atoms in its molecule. Deoxyribose means the same structure, but it lacks one oxygen atom in the structure. So it is called as deoxyribose. And both molecules are represented by the symbol you can see here as a hexagon. Now, what are the bases present in the DNA we can see. So the bases are differentiated into purines and pyrimidines. So adenine comes under purines and thymine comes under pyrimidines. And cytosine comes under purines and guanine comes under pyrimidines. Okay, so there are four nucleotides as we saw in the first slide, no? ATGC. So adenine and cytosine comes in purines and thymine and guanine comes under pyrimidines. Now this is the structure. So the nucleotide basically it contains a deoxyribose sugar molecule in the center and a phosphate group and one of the organic base. It may be adenine or guanine or anything, one of the bases. And these three structures they combine together to form a nucleotide. So a DNA is a molecule, is a long chain of a molecule of nucleotides. Okay, it is formed by millions of nucleotides which are joined together to form a long chain which we call as a DNA. So, in fact, the DNA usually consists of a double stranded of nucleotides. See here you can see it is a double stranded one. So, the outer strand it has sugar and phosphate chains and inside you have the bases. Let us see the arrangement now. So, as I already told, this is the deoxyribose sugar molecule which is attached to the phosphate group and one organic bases. It is a double strand. This picture shows the single strand, but DNA is a double stranded one, so it joins with the other nucleotides. So here, how it is joining with the other nucleotides, we can see here. So it is we call it as a Chargaff's rule. That means the bases always pair in the same way. How does it pair? So adenine forms a bond with thymine. Adenine always combines with thymine and cytosine always combines with guanine. This is the Chargaff rule. Okay, so adenine it combines with thymine and cytosine it combines with guanine. It is the Chargaff's rule. So bonding we can see here deoxyribose sugar, phosphate group and the organic base adenine. So adenine it combines with thymine, cytosine it combines with guanine. So can you say now, so thymine it combines with adenine. Yes, guanine it combines with cytosine. It forms the double stranded of a DNA. So this picture also clearly shows that DNA is a double stranded one. Now, here we can see that the DNA, the ends of the DNA are denoted as 5 prime or 3 prime end. We call it as 5 prime or 3 prime end or 5 dash or 3 dash end. Why it is differentiated like that? Because at one end, if you see the DNA molecule, at the one end of the DNA molecule, you can see the phosphate group attached to the fifth carbon of the deoxyribose sugar structure. In 3' end, you can see the attachment of phosphate to the third carbon group of the deoxyribose sugar structure. So only it is differentiated as 5' and 3' end. Is it clear for you? So the outside the strand it has deoxyribose and phosphate group. Inside you have the organic bases. So always adenine pairs with thymine and cytosine pairs with guanine and the bond between these bases are the hydrogen bond. Now, the paired strands are coiled into a spiral called double helix. We say that DNA is a double stranded and it's a double helix structure. What do you mean by that? We can see here. So it is made up of two strands. Helix means for every 10 nucleotides, it's spirally twisted. Okay, so only we call it as the double helix. So it always turns in right-handed direction. So you can see here clearly. See outside we call it as a sugar phosphate chain and inside we call it as an organic base. For every 10 nucleotides it get twisted. So only we call it as a double helix structure. So here we can have the major group and this one will be the minor group. Okay, the distance between the strands when they are twisting around the right hand side. Now, there are different forms of DNA. bDNA, aDNA, Z, Z, D, E DNA. and all organisms are made up of bDNA only. Basically, we have this bDNA. In some cases, a DNA or zDNA can be found. So, what makes the DNA different? That is nothing but it can be differentiated according to the turning of the strands, nucleotides. If it is turned in the right hand side, we call it as A or B form. Left hand side, it is called as Z form. In between A and B form, there will be slight differences. The base pairs, after 11 base pairs, it will be twisted in A form, and after 10 base pairs, it will be twisted in B forms. And the angle, there will be also some difference in that. So it differentiates the DNA as A and B form. Now, what do you mean by the genetic code? So, the sequence of the bases, we are learning no organic bases, adenine, guanine, cytosine, thymine. So the sequence of the bases in DNA, it forms the genetic code. Okay, genetic code means nothing but it is a triplet which codes for a particular amino acid in the cell. You can see here clearly. So DNA is a double stranded one. The triplet C, A, T, G. If you take these three bases, it codes for one amino acid. Then CTA, it also codes for a amino acid. GGC, it codes for another amino acid. So these amino acids combine together to form a protein. So the information for the protein formation is seen inside the DNA in the form of genetic code. You can see some example. Cytosine, adenine, thymine, it codes for valine. Cytosine, guanine and adenine, it codes for alanine, which is an amino acid. So these amino acids, they combine together to form proteins, which we all know that protein is very important for our body because proteins are the building blocks of our cell, and amino acids are building blocks of proteins. So amino acids are the smaller parts of the proteins. So the sequence of the triplets in the DNA molecule may code for a complete protein because it carries the information for the formation of a protein. So that sequence we call it as a gene. Okay, so the functional part of the DNA we call it as a gene which carries the information. So there are 1,000 and more number of bases in one gene. You can see here cell inside the nucleus we have the chromosomes DNA, and the DNA you can see the information present inside the DNA. The functional part of the DNA we call it as the genes. Information is that nothing but the portion which codes for a particular protein, we call it as genes. Hope you all understood about this. Good. So as a recap, we can see inside the cell we have nucleus, and inside the nucleus we have the chromosomes. And the chromosomes, a small part of the chromosome, we call it as DNA. The DNA has the sugar base and the organic bases inside that we call it as a nucleoblase or nucleotides. There are 4 different types: adenine, guanine, thymine and cytosine. So adenine and guanine comes under purines. Thymine and cytosine, it comes under pyrimidines, and the bonding between them is always adenine pairs with thymine and guanine pairs with cytosine, and the bond between them will be the hydrogen bond. Hope you all understood about the DNA molecule. Very good. Here in this video, I have posted 5 questions in this. So go through the video carefully and answer the questions and leave your answer in the comment section. Thank you for your listening and happy learning.